Thanks, Jignesh. Our next speaker is Dr. Stephen Schiff. He's a professor and vice chair for global health in the Department of Neurosurgery at Yale University. A pediatric neurosurgeon, he's got an interest in neural engineering and sustainable health engineering. He received his NIH Director's Pioneer and Transformative Award in 2015 and 2018, respectively. He's been working on disseminating a low-field MRI technology in low-resource settings. And this has led to the discovery and characterization of neonatal pan-bacillus, a new disease syndrome that is an important cause of infant sepsis and hydrocephalus in East Africa. And he's connected by Zoom here.
Well, thank you very much. Can you all hear me okay?
Yes.
Wonderful. So I really appreciate the opportunity to be part of your symposium today. And we think globally that there may be as much as 400,000 new pediatric cases of hydrocephalus each year. The vast majority of those cases are in the developing world. The greatest concentration of cases is in sub-Saharan Africa, and the largest contributor to those cases is following infection. So at this one site in eastern Uganda, the Cure Children's Hospital, since 2001, they've treated over 13,000 infants and children with hydrocephalus. Over 8,000 are post-infectious, and essentially all have been culture negative. As Dr. Taylor just showed you, if you put an endoscope into the third ventricle, our preferred way of trying to treat these children, then a typical appearance, I hope you can see my mouse here, a typical appearance will show an array of small pustules adherent and lining the ependymal surface in the ventricle. Often we come through the floor of the ventricle, as he described, and you see a relatively pristine space that doesn't look like it's had an infection before. We see this appearance in these children in East Africa, but if you walk into an operating room in Hanoi, you'd see often a very similar picture of adherent pustules, and my colleagues have seen the same thing in Haiti. It's a global phenomenon that we've not understood much about. I visited this hospital first in 2007, and I was interested in trying to do something helpful. I asked Ben Worf, who was the medical director at the time, what was the most important project I might be able to work on that they needed help with. And he said, well, maybe I could figure out what makes these kids sick. And I thought, how hard can that be? As a child, I had leafed through my grandparents' edition of the well-known De Kroof book on microbe hunters, and I figured, you bang on doors, you find people who know something about infections, and you figure out what the cause is. I'm here 16 years later, finally able to answer some of those questions. And our big break came with very substantial support from the NIH Director's Office. Solving this required a level of resource commitment beyond what we were able to ordinarily secure. It enabled us to use very high quality technologies for both for preserving these specimens on site at multiple places in Uganda. We set up a cryogenic infrastructure that was robust against the insecure electrical grid using liquid nitrogen. We used large commercial dry shippers and managed to ship over 8,000 frozen tubes without losing any to thawing from Africa to the United States. So what do you do if you're faced with children that have the clinical symptoms and signs of infection, but you can't grow anything, and you have nothing that comes up positive on your standard infection panels? Well, you can amplify a gene from bacteria, which humans don't have, and identify at least the genus of bacteria through sequencing that gene. But to identify viruses, fungi, and parasites, we've turned to deep RNA sequencing. And by 2020, in 100 post-infectious and congenital hydrocephalus patients, we use congenitals as controls. We started finding that one organism seemed to dominate these infections. It was a painibacillus, which is Greek or Latin for almost a bacillus, a genus that was described about 20 years ago and broken off from the bacillus family. It forms a highly destructive infection in the brains of these children. Here's a typical appearance with multiple calcifying abscesses. Everything I'll show you now will be on infants under 90 days of age. And it was extremely difficult to grow in culture. This species was Thiaminolyticus. So Painibacillus thiaminolyticus has been known since 1950 or so. It was first found in the gut of humans in Asia. It was not known to be a pathogen. If we place that reference strain into mice in their peritoneum, they don't get ill. If we take the African strains of that same species, they're dead within a day or two. We managed through over 1,000 different types of cultures on hundreds of these specimens to finally get 3 strains to grow. We assembled their genomes. We looked at their antibiotic sensitivities and resistance patterns. These organisms have picked up multiple antibiotic resistance genes. They also have picked up, in contrast to the reference strain, a type 4 pilus system. That's a little hair-like secretion system which helps certain bacteria form biofilms and adhere to things. And it is one of the virulence factors of Neisseria meningitidis. We are working below the meningitis belt that goes across Central Africa. We're about 200 kilometers below that. But the bacteria that we've found here shares a virulence factor with that organism, which is also highly neurotropic. If we CRISPR edit out that type 4 pilus system, the organism loses much of its lethality. But now that we've identified this, there are many unanswered questions. We don't know when this infection was acquired. We don't know if it even caused the hydrocephalus. It could be a secondary infection. And how do you solve this? Well, most of the mothers would report to us that their infant was born without a large head, had a very serious febrile illness, often with shaking and jerking of the limbs, suggesting seizures in the first few weeks of life. Then after the child had survived that, the head began to grow. If this sounds like neonatal sepsis, and worldwide neonatal sepsis is still killing upwards of 1 million infants a year around the world. That's many more deaths than are attributed to malaria of any age. I've testified to Congress about this situation. We don't know what causes most of the cases of neonatal sepsis worldwide, and we've ignored it as a medical community, both from research and funding agencies, as well as government and World Health agencies. As I mentioned in my testimony, newborn infants dying in the developing world have never had a political voice. More recent wonderful studies, I especially like the Cambodian study and the ANISA study, the large South Asian study, with very, very good quality, are still identifying perhaps 2% of the putative pathogens in global sites for neonatal sepsis. So what did we do? We set up 3 linked simultaneous case control trials in Uganda. 1,400 subjects, over 8,000 samples. We then confirmed with both genus and species this particular bacteria using over 2,500 qPCR reactions. This just came out in the Lancet Microbe. And so we work backwards. We start from hydrocephalus. We know that a lot of them may have come from neonatal sepsis. Many children with sepsis have a perinatal transmission. We're also concerned with environmental sources. So let's start by comparing post-infectious hydrocephalus and congenital hydrocephalus without a history of infection. Almost half of these post-infectious cases are PCR positive on admission for Painibacillus. We ran a maternal trial, and this is laboring mothers giving birth both in the east and the western regions of Uganda. Half of the mothers were septic at the time of presentation. Not a single DNA read to Painibacillus in samples from their birth canals, placenta, maternal blood, and cord blood. Let's look at neonates with sepsis. 6% of 800 patients with samples of blood and spinal fluid from east and west in the country were positive for Painibacillus as a source, as a cause of their infection. Here's one piece we were really looking for, was linkage. Could we find neonates with sepsis that recovered from the sepsis and then came back with the same infection with hydrocephalus? These are two cases. They're 1-week-old infants. Note in this child who has both 16S evidence and then confirmatory PCR evidence of Painibacillus thiaminolyticus. Note the little cyst in the frontal lobe when septic. This child gets 3 weeks of ceftriaxone, which turned out to be a very good choice for this organism based on laboratory sensitivity. Child is afebrile, feeding well, looking alert, goes home, comes in a month later with substantial hydrocephalus. And now note that that small cyst has turned into a maturing, calcifying abscess in the frontal lobe. PCR again confirms the presence of the same organism, which was not sterilized in the nervous system on 3 weeks of antibiotics. Here's another case. This child has extensive white matter destruction, comes in at a week of age, again 3 weeks of ceftriaxone, returns 3 months of age with heavily destroyed frontal lobes. Note the increased density of this lobe. This child also remains PCR positive for Painibacillus. So then we looked at the disease syndrome in the neonates. And we've named this a new disease syndrome. There's really no literature on this at all. We call it neonatal painibacilliosis. It is a very virulent infection. 14% of those children went on after treatment and survival to get post-infectious hydrocephalus, almost unseen in any other organism causing neonatal sepsis in this cohort. But it's misleading. You have to survive in order to get post-infectious hydrocephalus. More children died during excellent treatment than went on to get post-infectious hydrocephalus. So it's a highly lethal and very and destructive infection, both of the brain and of the lives of these children. We just reported this in Clinical Infectious Diseases. There's a very nice editorial by Michelow and Obara in that same edition of the journal, talking about the critical need for better tools for us to diagnose and understand neonatal sepsis. And then it turns out you have to know the PCR status at surgery. We either do shunts, as Dr. Taylor said, or we try to do endoscopic surgery. Look at the results if you're PCR positive of the ability of the diversion procedure, shunt or ETV, to function. If you're PCR positive, it destroys the function of the endoscopic third ventriculostomy. So we have to learn how to treat this at the surgical site when patients come in for care of their hydrocephalus. Most recently, since we've been publishing about this organism in Africa, we're getting multiple case reports appearing now in the United States. Same organism. It's Paenibacillus thiaminolyticus for the ones I'm showing you here. There are a few cases of a related species, Dendritiformis. These cases have the same destructive brain appearance that we see in the African patients on the top row. These are US patients in the last 2 years, and most of the US patients, despite good US care, have died as well. Going forward, how do we handle such things? So we know a lot about the patterns in Africa, but I've just spent years, seriously years, giving you the molecular diagnostic conclusions from these 1,200 patients. So with support from the NIH Director's Office for what I called predictive personalized public health, we're trying to look at the patterns to give us a better way at point of care to create our differential diagnoses and better treat these children. Here's the spatial concentration of where post-infectious hydrocephalic cases are coming from in Uganda. This is over 4,000 geolocated cases. Here's Lake Victoria. These are the northern banks of Lake Victoria, a very swampy, wet area in a plateau. This is Lake Choga, where the Nile flows in and then out of, again with huge swamps on the southern and northern banks. And this is where this organism is concentrated, as well as piling up against the Kenyan border. We've taken all 60,000 villages in Uganda and placed them onto the satellite climate grids. We're looking at vegetation indices, and I'll explain that in a minute. If we look at the rainfall in each patient's village when they're born and then go backwards a year, what we find is there's a tremendous association. When the rains come in just before birth, this organism becomes a high risk for these infants. This is a village, Kihadi, in eastern Uganda, where one of our patients had come from. Dr. Malando here and I are spending a few hours talking with the villagers, trying to decide as soon as we get our approvals, which we're starting to get in now, how we're going to conduct the field work needed to identify this organism in the environment. We've worked very hard with our African collaborators and stakeholders to negotiate a genome and data sharing policy. One of the things we've done with this is looked at the ancient migration patterns into this area of Central Africa. Really, there's two large migrations that contribute to the population in the modern nation state of Uganda. There's a Nilo-Saharan migration up to about 1,500 years ago. And then there's the Bantu expansion from Southern Africa, which came in starting about 5,000 years ago. If you do whole genome sequence analysis of 1,000 of these infants, you see these migration patterns readily evident, painted onto the geography of the country. We're presenting this today at the Human Genetics meeting in Washington, D.C. So here's the rainfall pattern for post-infectious hydrocephalus. Spina bifida is the next most common source of hydrocephalus in this population. Look, this is individual village averages for rainfall. The children with infection come from wetter villages compared to the children with spina bifida. There's no association with spina bifida at the time of birth, but we're vigorously looking at other correlates that may have a role. Vegetation for a nutritionally sensitive malformation, spina bifida, the poverty index, and interestingly, there may be a link to ancestral admixture. Now, most brains don't have access to imaging around the world. We have placed a low-field commercial MRI scanner at the hospital in Mbali during lockdown. Most recently, we've done an assembly of what we think is the next generation of such sustainable low-field imagers. This is an engineering group that I collaborate with in Mbarara, Uganda. This was recently written up in both Nature and Science, and we recently wrote a nice review of this type of next-generation technology. What do you use it for? As Dr. Taylor very nicely told you, an endoscopic case can leave you with large ventricles. These are 2 post-operative cases. And usually we look at the thickness of the mantle to determine if we've decompressed the brain well, but it can be very misleading. This child has a small brain, and this child is not doing well on developmental testing. This child has a brain of almost normal size, riding on those larger ventricles, and this child's development is almost normal for age. So we drain fluid, but we're in the brain growing business. And there is beautiful old data looking at how the growth of the brain relates to body size. That's called allometry, the different proportions of the body, and it has a very unfortunate history. Paul Broca, whom some of you recognize as a neurologist in the 19th century, did a lot of work on this. He was doing it for some motivational reasons that were based on both looking at males versus females and different nationalities in Europe to prove who might have been best. Stephen Jay Gould wrote a wonderful book about this and looked at Broca's data. We looked at over 1,000 scans of normal North American children. Broca's difference in brain size between normal children and then young adults is easily seen. Gould's difference, looking at normalization of brain size by body size, is still reproduced here. Unexpectedly, we found that the ratio of brain to fluid in the normal child is very tightly regulated. We don't understand what does this, but you don't have to look at whether a child is male or female or large or small. That fluid, which is floating your brains right now while you listen to me, is extremely tied to the actual size of the brain throughout the growing child's life. So we've put together normal brain growth curves for the normally developing child, and we use those curves in our clinical trials, our randomized trials of surgical techniques for hydrocephalus to ask how well we're doing in managing that brain growth. We do this in our ongoing randomized trial entering its 10th year of follow-up in Africa. And as Dr. Taylor mentioned, we're part of the ESTHI trial, and we're also using brain growth as a metric for success in the North American ESTHI trial. We're putting technologies for imaging into parts of the world without any radiologists. So we're trying to embody the expertise of a radiologist into a variety of advanced machine learning algorithms to be an assist for the physicians on site with these new imagers. And nobody does this alone. I've had the tremendous privilege of working with some of the best people on earth who've joined us to try to help these kids. And they've really poured their hearts and souls into this. And everything I've told you today has been supported strongly from NIH. So I'm delighted to answer some questions. I really thank you for your attention today.
Steve? Could you, Dr. Schiff, stop sharing, please?
Yes, I can.